This conference will now be recorded.
So today we'll be dealing with what is the mechanism of action of insulin and glucagon. So first we'll just see what is the history regarding insulin. So the word insulin was derived from the Latin word insula, that means island or an islet. Then in 1869, Langerhans was a scientist who identified the alpha and the beta cells in the islets of pancreas. And in 1922, Banting and Best, they were the two scientists who extracted insulin from the pancreas. And insulin was the first hormone to be isolated in a pure form. And the experiment that was done was that they had done a pancreatectomy for an animal, and they started giving insulin so that the survival of that animal increased when they injected insulin. In 1927, he crystallized the insulin, and in 1954, it was Sanger who studied the amino acid sequence of the insulin, and it was therefore the first protein in which the complete amino acid sequencing was done. Then, for this work, Sanger got the Nobel Prize in 1958. Then, insulin was also the first protein produced by recombinant DNA technology in 1982. So you can see that insulin has a lot of first. So it was the first hormone to be isolated. It was the first protein to be undergo complete amino acid sequencing, and it was the first protein also to be produced by recombinant DNA technology. Then how does the structure of insulin look like? So as you know, the insulin consists of 51 amino acids, A chain having 21 amino acids, and the B chain, it has 30 amino acids. These two polypeptide chains, they are linked by disulfide chains as well as interdisulfide as well as intradisulfide linkages. So in interdisulfide, there are two linkages that is at A7 to B7, and then another linkage between A20 to B19. While the intradisulfide linkage is between the 6th and the 11th amino acid in the A chain. Now, if you see for how the insulin biosynthesis takes place, so the insulin is first synthesized as a pre-pro-insulin. So this you can see, this is the structure of pre-pro-insulin, which consists of 109 amino acids. This then from this pre-pro-insulin, a signal sequence of 23 amino acids will be removed, and this will take place in the endoplasmic reticulum. So after removal of the signal sequence, it will result in the formation of a pro-insulin consisting of 86 amino acids. Now from the pro-insulin that is synthesized in the endoplasmic reticulum, it is transported to the Golgi apparatus, where it will be cleaved by an enzyme that is a protease to form one C peptide. So you can see the in between peptide that is removed is the C peptide, and the remaining two chains become the A and the B chain. So you have insulin, and then you have the active form of insulin, and then the C peptide. Now, once insulin, the active form of insulin is synthesized, it is packed into granules, and the molecules, they take the shape of a hexamer with 2 zinc ions and 1 calcium ion. Now, the proinsulin will have only about 1/3 the biological activity of that as that of insulin. While insulin and C-peptide, they are ultimately synthesized and secreted, they will be in equimolar quantities. Therefore, the measurement of C-peptide is an index of the rate of secretion of insulin. Actually, C-peptide as such does not have any function. The importance of C-peptide only is that if you quantitatively measure the C-peptide, it will give you an index of the rate of secretion of insulin. Then, what are the factors that affect the insulin secretion? There are factors that may increase the insulin secretion or they may decrease the insulin secretion. Now, glucose is the major stimulant of insulin secretion. because as the blood glucose level increases, then insulin secretion also correspondingly it increases. So you can see here here there is a glucose entry occurs through the glucose 2 receptors in the beta cell of the pancreas. So here as the glucose, more glucose enters, more ATP synthesis takes place. So what will happen? The ATP channels, potassium channels, they close. Once ATP increases, the potassium channel closes. When the potassium channel closes, then there will be entry of calcium into the cell. This in turn will cause the release of insulin from the stored insulin granules. Now the insulin that is secreted, it is this glucose that will ultimately lead to the secretion of insulin. It is a biphasic response will be seen in the insulin secretion. So what do you mean by that biphasic response is that initially there is a rapid secretion of insulin for the first 2 minutes. Later on a second phase of insulin secretion will be there that will last for about 5 to 10 minutes. But the secretion of insulin will be less, and it indicates that the second response to insulin secretion means a new hormone is being synthesized. Then amino acids also induce the secretion of insulin, because after heavy ingestion of a protein, the amino acids, there is a transient rise in this amino acid concentration. And among the amino acids, arginine and leucine, they are the potent stimulators of insulin release. Then you have the gastrointestinal hormones, secretin, gastrin, pancreazymin. They also enhance the secretion of insulin. Then these GIT hormones also are released after the ingestion of food. So you have factors like glucose, which is the major stimulant for the release of insulin. Then amino acids after ingestion of protein-rich milk. Then once your food is ingested, there is also the release of gastrointestinal hormones that enhance the secretion of insulin. Then other than that, you have factors that inhibit the insulin secretion. So you have epinephrine, especially epinephrine during the stressful conditions or during exercise, the adrenal medulla will release the epinephrine. And this epinephrine is a potent antagonist of insulin. So it suppresses insulin release, and at the same time, it mobilizes also glucose from the liver to meet the energy demands. Then alpha adrenergic stimulation also leads to inhibition of insulin secretion. So then degradation of the insulin takes place in the liver. The plasma half-life of insulin is only 5 minutes, and a specific protease is there, insulinase, that leads to degradation of insulin. Then how does this active insulin, once insulin is synthesized, how does it act? So we said these are the factors that increase insulin secretion. There are the certain factors that decrease insulin secretion. So once insulin is released, how does it act? So insulin has to go and bind with the insulin receptor that is present in the plasma membrane to initiate its action. So this is the picture of insulin receptor that you can see here, and there will be insulin receptor is a polypeptide consisting of, it is a glycoprotein consisting of 2 polypeptides, and it has 2 alpha subunits and 2 beta subunits. The beta subunit will have tyrosine kinase activity. The alpha subunit is present, you can see, to the other side of the external to the plasma membrane, while the beta subunits are towards the cytoplasmic site. So once when the insulin goes and binds with this alpha subunit, what will happen is that the beta subunit gets stimulated. It transmits the signal to the beta subunit in the cytoplasm. And here the tyrosine kinase, because of the activity of the tyrosine kinase, it gets activated, and it gets autophosphorylation of this tyrosine kinase units occur. This tyrosine kinase units that are present on the beta subunit, they get autophosphorylated. This, in turn, this phosphorylated tyrosine kinase, what they will do, they cause phosphorylation of the insulin response substrate. So you can see here, this in turn gets phosphorylated. Once this IRS or insulin response substrate is activated, it will lead to a series of events that are known as cellular responses. Then insulin also acts at the transcriptional level to regulate the synthesis of more than 100 of proteins. So some of the proteins that it induces, and some it causes repression. So there's some example of the protein that it induces is glucokinase, pyruvate kinase, phosphofructokinase, and acetyl-CoA carboxylase. Then insulin represses the enzymes like glucose 6 phosphatase. These are the enzyme of gluconeogenesis, phosphoenol pyruvate carboxykinase, and fructose 1,6 bisphosphatase. Insulin also causes covalent modification, and through covalent modification, that is phosphorylation or dephosphorylation, it will either increase or decrease the activity of certain proteins or certain enzymes. So here you can see this is an example we have given of where all this enzyme, glycogen synthase, pyruvate dehydrogenase, pyruvate kinase, acetyl-CoA carboxylase, and HMG-CoA reductase, their activity is increased by dephosphorylation. So this is through a method of covalent modification. And insulin activates this enzymes to dephosphorylation. Then through this IRS pathway or signal transduction pathway, what we have just seen, insulin also increases the DNA synthesis, the cell growth, and anabolism. And glucose uptake, it is increased because insulin increases the recruitment of glucose transporter 4 in the cells. Then what are the actions of physiological actions of insulin? Now insulin, it regulates the metabolism of carbohydrate, lipids, and proteins. So how does it do so? We'll be seeing now. So the uptake of glucose, we have just said that insulin facilitates the uptake of glucose by recruiting this glucose transporter. So insulin facilitated diffusion of glucose in the muscle, and the transporter, this glucose 4 transporter, this is insulin dependent. So it stimulates this glucose 4 transporter. Glucose transporter 4, while glucose uptake in the liver by the glucose transporter 2 will be independent of insulin. So in the muscle, it is insulin dependent. In the liver, it is insulin independent. Now here, this is a picture showing the translocation of glucose transporter 4 from intracellular vesicle to the plasma membrane under the influence of insulin. So here you can see this is the glucose transporter, and this is the clathrin coated vesicle. So there is internalization of this early endosome, resorting, and you can see this is the glucose 4 transporter that will carry the glucose. Then once it delivers glucose, again it will go back to the plasma membrane. Then utilization of glucose. So the utilization of glucose occurs through the action of insulin. So glycolysis is increased. So insulin stimulates the glycolytic enzymes like glucokinase, phosphofructokinase, and pyruvate kinase. While it also favors glycogenesis, so it will stimulate or increase the glycogen synthase enzyme activity. Then insulin also promotes lipogenesis. So that's why it will lead to stimulation of acetyl-CoA carboxylase enzyme and glucose 6 phosphate dehydrogenase enzyme, which is NADPH dependent. So through insulin, it stimulates glycolysis, it favors glycogenesis and lipogenesis. Then the other actions of insulin is that it decreases glucogenesis by inhibiting or decreasing the key gluconeogenic enzymes. So these gluconeogenic enzymes are pyruvate kinase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. Then insulin, it prevents lipolysis by decreasing the levels of this hormone sensitive lipase enzyme. Then it also prevents ketogenesis by inhibiting or decreasing the HMG-CoA synthase enzyme activity. It prevents glycogenolysis, and by inhibiting the enzyme glycogen phosphorylase and glucose 6 phosphatase. The other effects of insulin is that insulin is also an anabolic hormone. So the protein synthesis will be promoted and degradation will be decreased. The insulin, it stimulates the replication of the cells, and in fact it has been said that insulin is an essential growth factor for all the mammalian cells. Now, what if you see in general, what are the other biological effects? What we have just seen now, we will summarize them. So we said, what is the effects of insulin in the carbohydrate metabolism? It will favor glycolysis by stimulating the enzymes of glycolysis, that is glucokinase, phosphofructokinase, and pyruvate kinase. So overall, what it's doing, overall effect is hypoglycemia. Then it depresses or inhibits gluconeogenesis by inhibiting the enzymes, that is pyruvate kinase, again pyruvate carboxylase, pyruvate phosphoenol, pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. And overall effect is that it promotes hypoglycemia. Then it promotes glycogenesis. Therefore, it leads to activation of glycogen synthase. It disfavors glycogenolysis. So it is inhibiting the glycogen phosphorylase enzyme. So overall effect is that it decreases the blood glucose levels. Then it also causes stimulation of glucose 6 phosphate dehydrogenase enzyme, and it leads to generation of NADPH, and overall effect is it leads to lipogenesis. Then what is the effect on lipid metabolism? So it causes stimulation of acetyl-CoA carboxylase and glycerol kinase, favoring lipogenesis, while and lipolysis is inhibited because it inhibits the hormone sensitive lipase enzyme, and it stimulates again cholesterol synthesis by promoting HMG-CoA reductase enzyme activity. On the protein, we said it has generally it has an anabolic effect, and it inhibits catabolism. So it causes inhibition of transamination and ornithine transcarbamylase enzyme, while RNA polymerase is favored. Now glucagon. So we have finished with insulin. We will move on to glucagon. So glucagon is a peptide hormone with 29 amino acids, and it is secreted by the alpha cells of islets of Langerhans in comparison to insulin, which is secreted by the beta cells of the islets of Langerhans. Now the half-life of glucagon is only 5 minutes. It is inactivated in the liver, and the major regulator of the secretion of glucagon is glucose. And because an increase in blood glucose level will inhibit the secretion of glucagon. So while an increase in blood glucose level will lead to secretion of insulin, an increase in blood glucose level will inhibit the secretion of glucagon. So how does this glucagon act? So glucagon the actions of glucagon will be the opposite to that of insulin. So while insulin favors glycogenesis, glucagon, it stimulates phosphorylase, glycogen phosphorylase enzyme. Therefore, it is promoting glycogenolysis. Then it also promotes lipolysis by stimulating the hormone sensitive lipase, while you can see that it inhibits glycogen synthase and phosphofructokinase, and it also inhibits acetyl-CoA carboxylase. Then glucagon also favors gluconeogenesis, and it activates the enzyme fructose 2,6 bisphosphatase. Then it also promotes beta oxidation by stimulating the enzyme carnitine acyltransferase enzyme. But how does this glucagon enzyme act? Glucagon hormone acts. So like insulin, glucagon also, it has to bind with the receptor on the plasma membrane. Now this receptor contains a GDPG protein, which is in an inactive form. So this GDP is exchanged for GTP so that the G protein becomes active. This activated G protein, in turn, it will stimulate the adenyl cyclase enzyme. So this adenyl cyclase enzyme, once it is activated, it will cause the synthesis of cyclic AMP from ATP. So adenyl cyclase converts ATP, stimulates the conversion of ATP to cyclic AMP. This cyclic AMP in turn binds to the regulatory subunit of protein kinase and releases the catalytic subunit of protein kinase for phosphorylation of the enzyme proteins for their action. Now, if you compare the differences between insulin and glucagon, so you can see here that insulin promotes glycolysis while glucagon inhibits it. Glycogenolysis is inhibited by insulin and favored by glucagon. Gluconeogenesis, it is inhibited by insulin, it's stimulated by glucagon. Glycogen synthesis is activated by insulin, inhibited by glucagon. Then lipolysis and ketogenesis, both are inhibited by insulin, while both will be promoted by glucagon. Then protein, insulin promotes anabolism of the protein, while glucagon favors catabolism. So ultimately, what happens to the blood glucose level? Blood glucose level is decreased by insulin. Blood sugar levels are decreased by insulin, and glucagon leads to increase in blood glucose levels. So this combined action on insulin on glucagon will help to keep the blood glucose within normal limits. So high blood glucose will stimulate the blood glucose insulin secretion, while low blood glucose level will lead to secretion of glucagon, and it leads to increase in glucose production. So if you, besides glucagon, there are also other anti-insulin hormones like glucocorticoids and growth hormones. So we will just see, we have already seen the comparison between insulin and glucagon. We will see what is the effect of glucocorticoids and growth hormone. So you can see that glucocorticoids, like glucagon, they stimulate gluconeogenesis. So both glucocorticoids and growth hormone, they favor gluconeogenesis, and they also favor lipolysis. Growth hormone also leads to ketogenesis. So that means, like glucagon, they increase the blood glucose levels as compared to that of insulin. So all these are glucagon, glucocorticoids, and growth hormone. They are called as anti-insulin hormones.